Hello, everyone. Our chapter for today is about types of gene mutations. Already, we talked before about gene mutation, which is an error at the level of the DNA molecule sequence of nucleotides. But our objective for today is to find the types of these mutations or the types that may hit the sequence of nucleotide. Let's remember together what do we mean by a gene mutation? Gene mutation is once the normal gene, which is responsible for synthesis of a normal protein, is hit and it becomes an abnormal one. So, this is a mutated gene or an abnormal gene or a gene that underwent certain mutation. An error takes place in its sequence of nucleotide. As a result, an abnormal protein is produced, or maybe no protein is produced. This is a reminder about a gene mutation. Now we have to talk about the first type of gene mutation. Let's take into consideration the first type to study. Our example for today or for this type is the sickle cell anemia. Already you know now, sickle cell anemia is a disease and it's because of an abnormal synthesis of hemoglobin. Here you have a normal hemoglobin in a normal red blood cell, and the function of this protein is correctly taking place, in opposite to this one, where the abnormal hemoglobin is produced and the sickle cell is produced now, and an abnormal function takes place. Okay. Let's compare me and you together now. Let's compare the sequence of a given nucleotide in this gene responsible for the synthesis of this protein. So, we have CTGGAG. If I compare it to the sequence which is responsible for the production of abnormal, we will find it's exactly the same except at the nucleotide where A is replaced by a G. So, in this case, again, let's go for comparison. Now you know that the sequence of nucleotide of a DNA is responsible for transcription or transcribing a messenger RNA, which in turn is responsible for the synthesis of protein by finding the certain sequence of amino acids. Okay, so in the sickle cell anemia, this is the sequence of the normal DNA molecule, and this is the sequence of the abnormal one. Again, with comparison, you will see both have exactly the same sequence, except that of the fifth nucleotide, where T here is replaced by A. Okay, so let me find the type of this hit or this error or this mutation. So it is a mutation for sure because an error occurs. So the mutation occurs only at one point, which means only one nucleotide is replaced. That's why we say it's a point mutation. Okay, it's a point mutation because it's at one point. But what had happened at this point? There's a kind of replacement, or in other words, substitution. The T is substituted by A, and once it's substituted by a different sequence occurs and a different result takes place. So, the messenger RNA here, if you have to compare its nucleotides also, you will see it's exactly the same except at this nucleotide where A is replaced by U. and as a result, a glue as an amino acid is substituted by val or valine as another amino acid. So it's a mutation, it's a point mutation, it's a point mutation by substitution. What is the effect of this mutation? And this is the very important thing here. So, because we have an altered polypeptide, a changed one, we have an abnormality, a different function of the produced protein, we have an abnormal hemoglobin here. So, it is a missense mutation, it is an altered polypeptide, so it has an effect which is alteration of the protein. So it's a missense mutation. Here another effect of certain substitution. Let us find together or put a question in our mind together. Is any substitution responsible for giving this alteration? We don't know. That's why we are going to take another example. Here, a sequence of a nucleotide of a normal DNA molecule without any mutation. And here is the sequence of a nucleotide of a gene which is hit by mutation. First of all, we have to compare. Let us compare the sequence of nucleotides of the DNA. All have the same sequence of nucleotide except A here is substituted or replaced by T. Okay, so now we have to go to find the types of amino acids that are produced now. So the first amino acid is methionine. Methionine in both. So CCA codes for pro. CCT also code for pro because as you know, in the genetic code table, there are common sequences that give same code for the same amino acid. And the last one is serine and serine. So methionine, proline, serine, methionine, proline, serine. So although there is a mutation at the level of DNA, we didn't observe any change at the level of amino acid. Both codons, they code for the same type of amino acid. So it is a substitution. Yes, it is a point mutation by substitution. It is an altered DNA, but what about its effect? The effect of this mutation is silent. What do we mean by silent? We mean that the error takes place and it occurs but silently without any detectable change. So, what's the type of this mutation? It's a silent mutation, for sure, it's substitution, point mutation by substitution, but it's silent. Why silent? Because it doesn't cause any change at the level of the protein. The protein remains functioning in the same way. Now, what about another effect? Okay, here I have a sequence of nucleotides which is also for or it belongs to a normal DNA molecule. And here we have the protein which is synthesized out of it. For sure, there is a messenger RNA in between, but we are skipping now this step just for simplicity. Here is the mutant DNA. Just let's take an idea or take a look about the result. So, the amino acids that are produced, histidine, cysteine, tyrosine, and glycine. What about the protein or the amino acid sequence which comes from the mutant DNA? So, where is the mutation? Here is the mutation. T is replaced by A, substituted by A. So it's a point mutation by substitution. But let us look for the effect. So here cysteine is coded, but TGA as a sequence or as a codon on messenger RNA, it doesn't code for any amino acid. It is a stop codon. So, because it's a stop codon, there is a termination as you see here. So, no more synthesis of this protein, and it stops with histidine in this case. So, that's why we are going to call it's a nonsense mutation because it gives an incomplete polypeptide. Let's make a revision or a summary for the different types of point mutations that occur by substitution. Again, point mutation only it takes place at one point. Here, when there is no mutation, TTC, messenger RNA AAG, and it codes for lysine. If the point mutation is silent, C is replaced by T, codes for AAA, but still giving glycine. So that's why no effect, it's silent. Nonsense when it codes for stop termination of the synthesis, and missense, okay, when it gives another amino acid, and in this case it altered the protein and it has a different function or it is not functional yet. Now let's go to the second type of types of mutation, and to put it under our study. Look here. So here we have the first sequence of nucleotides, and as you see, we start with an amino acid. We are studying a DNA, his, and it continues in this way. Okay? So if I have to compare the normal DNA to the mutant, the one which is hit by mutation, you will see. So CAT, while here we have CAT, A in addition, then CAT. So, what had happened here? What happened is an extra nucleotide is inserted between T and C. So, when an extra nucleotide is inserted between these two types or these two nucleotides, eventually what will happen? What will happen, because we are studying codon by codon, this inserted nucleotide will take with it the next C and the next A in order to be read together, as if as this A pushes all of the other nucleotides to the right. So the new translation will be ACA will code for threonine instead of Hess, and TCA now for serine, and TCA for serine. And as you see, all the coming amino acids changing, not only one amino acid changes. Why? Again, because this inserted amino acid pushes all the other, and it changes the arrangement, as if as it rearranges the codons, because we read triplets 3 by 3. So T, which is read here by CAT, now it's going to be read with the neighboring one. And the type of this mutation is insertion, where its effect is frame shift. What do we mean by frame shift? As if as we shift all the other nucleotides to one side, which is to the right here. And for sure, the polypeptide is altered and it has a different function because we have a sequence of amino acids with changes in this way. The third type under study is, again, we are going to take an example, DNA of a normal, DNA of the mutated one. What happened here? Again, CAT coding for histidine, his, until we reach this one here. Look, CAT. We will see that CAT, C directly TC. What happened to the A which was found here? It is deleted. A is deleted. It's not found in the sequence anymore. For sure, it's an error. We can't remove, we can't add any extra nucleotide. So, again, once this A is removed, eventually what will happen? T and C will come to this side and will be read together as CTC, again because they are read as triplets, as codons, and all the coming or the neighboring ones to the right will move to the left because the T will take the place of A and C take place of T and etc. And now instead of CAT, we have to read CTC, and instead of his, we have leucine. So the third type to study for the mutation is deletion, when a single nucleotide is deleted, and for sure it causes a difference, a change at the level of the sequence of amino acids, and the effect is also frame shift, but in this way they are shifted all of them to the left instead of to the right, or unlike the insertion which shifts them to the right. But also as a result, we have an altered polypeptide. So the three types of mutations are by substitution, by insertion, and by deletion. These are the three ones that you have to study. Finally, an application with you. I'm going to ask you to compare, how to compare the sequence of nucleotides. What you have to say is like this. Go to the sequence of a nucleotide and you will see that, or say that in both DNA sequences, the original, normal, and the muted one, the sequence of nucleotide is the same except one, which is number 4, where G is replaced by T in the mutated one. At the level of the messenger RNA, you will say the same. You will say the same. Same sequence of nucleotides, except in position 4, where C is replaced by A. At the level of amino acid, also you will say the same. Both have the same sequence of amino acids, except amino acid number 2 is replaced by serine, where arginine is replaced by serine. So, here is a way of comparison by numbering the nucleotides. Also, you can compare using triplets or codons, as you can say, in the messenger RNA, both have the same sequence of nucleotides, except the first nucleotide in codon number 2, which is C, is replaced by A. So this is another way of comparison. What you have to do is to follow your given and to compare in this way. What about the type of mutation that we can conclude here? Okay, for sure, it's a point mutation by substitution, and it is missense, since arginine is replaced by serine. Okay, I'm done here, and thank you very much.